Mitosis is used in the reproduction of somatic cells or body cells. Mitosis occurs only in eukaryotic organisms. The sequence of events is divided into the stages of prophase, metaphase, anaphase, and telophase. In addition, mitosis is preceded by interphase, when the cell prepares to divide. A majority of the cell cycle is actually spent in interphase. Ultimately, mitosis ends in two daughter cells which are genetically identical to the parent cell. Both daughter cells are diploid. In other words, they contain a full set of chromosomes. Mitosis varies in different species based on the number of chromosomes in a particular species' full set. This is an image of prophase of mitosis, used to exhibit each component of the cell involved in the cell cycle. So let's put mitosis into a perspective anyone can understand, using candy. For our demonstration,
Twizzlers will represent chromosomes,
and sour straws will be used for spindle fibers,
Skittles will create the plasma membrane,
Jolly Ranchers will act as the centrioles,
M&Ms will serve as kinetochores,
and pink sour straws will represent the nuclear membrane. Now let's take a look at the process of mitosis. Interphase is the longest stage of the cell cycle. It consists of gap 1 phase, where cell growth occurs and the cell decides if it is going to complete mitosis, S phase, when DNA is replicated, and gap 2 phase, in which the cell continues to grow and prepares to enter mitosis. During prophase, the nuclear envelope disassembles, the chromatid develops into the shape of chromosomes, and the spindle apparatus forms. This particular cell contains 4 duplicated chromosomes, each of which contains 2 sister chromatid joined together at a centromere. At metaphase, the 4 chromosomes line up at the metaphase plate, an imaginary line along the center of the cell. Here, spindle fibers fully attach to the kinetochores. Each chromosome contains 2 kinetochores, one for each sister chromatid. During anaphase, the spindle fibers begin to separate the sister chromatids at the centromere, pulling each chromatid to opposite ends of the cell. The sister chromatids are now considered individual chromosomes. The last stage of mitosis is telophase and cytokinesis, when the chromosomes begin to unravel and a nuclear envelope forms. A cleavage furrow develops and 2 daughter cells begin to take form when the cytoplasm divides through cytokinesis. The 2 cells that result are diploid, each containing a full set of genetic material. They are genetically identical to the parent cell.
Meiosis differs from mitosis in a few key ways. Meiosis is used to create gamete or sex cells used in the reproduction of eukaryotic organisms. Rather than creating 2 daughter cells, meiosis results in 4 daughter cells, all of which are genetically different from each other as well as the parent cell. In addition, each of the 4 cells are haploid and can only contain half of the genetic material of a cell created through mitosis. Meiosis consists of meiosis 1 and meiosis 2. Prophase 1 accounts for genetic variation of offspring. In this step, homologous chromosomes are visible inside the nuclear membrane, each consisting of 2 sister chromatid joined at a centromere. Different from mitosis, however, this step includes synapsis, or the joining together of pairs of homologous chromosomes forming a tetrad. This occurs when sister chromatid cross over at the chiasmata and results in the mixing of genetic information. During metaphase 1, each tetrad lines up along the metaphase plate, creating independent assortment, and spindle fibers begin to attach to the kinetochore of each homologous chromosomes. Next, during anaphase 1, spindle fibers separate homologous chromosomes by pulling apart each tetrad at the chiasmata. In this particular cell, there are now 2 sets of homologous chromosomes at each end of the cell, which vary from each other genetically. During telophase 1, a cleavage furrow develops and the cytoplasm divides into 2 new cells. A nuclear membrane forms again around the homologous chromosomes in each cell. These 2 resulting daughter cells are still diploid, containing a full set of chromosomes, because the sister chromatid are still attached at the centromeres. Following prophase 2 is metaphase 2, when homologous chromosomes line up along the metaphase plate and spindle fibers attach to the kinetochore of each sister chromatid.
Next, in anaphase 2, spindle fibers attached to each kinetochore separate sister chromatid by pulling them apart at each centromere. 8 total sister chromatid, 2 in each daughter cell, travel to opposite ends of the cell. The last stage of meiosis is telophase 2. Here, a cleavage furrow develops in each cell, and the cells proceed to divide into 4 new daughter cells. A nuclear membrane forms again around the genetic material in each of the 4 cells. Because duplication of genetic material did not occur before prophase 2, these new cells are haploid. They only contain 1/2 of the genetic material of the parent cell. Also, the 4 daughter cells are genetically different as a result of the crossing over that occurred during prophase 1.
Thanks for watching!